Ladies and gentlemen, my name is Yicheng from National Yangming Jiao University and the Guangzhou Hospital, Taipei Veterinary General Hospital Branch, Taipei, Taiwan. Thanks for the LRC Scientific Committee to inviting me for sharing our study today. My topic is Flexibility of Physio-Cognitive Decline Syndrome, a longitudinal cohort study. It's my honor to be here. At first, I will introduce the Physio-Cognitive Decline Syndrome, which we called PCDS. Due to declining physical and cognitive functioning, play critical roles in aging, and is also a clinical marker of incident disability, dementia, and mortality. So in our study, we define PCDS as simultaneous presence of both physical and cognitive impairments, but no dementia. The longitudinal transition of PCDS phenotypes remains unclear, and the concepts of cognitive frailty is widely accepted in different research groups, but the name and the diagnostic domain of physical and cognitive parts is different. For example, in the table below, the physical parts include physical frailty and pre-frailty in cognitive frailty. And in motoric cognitive risk syndrome and RPCDS, we use slowness and oral weakness. And in the cognitive parts, the subjective and objective cognitive impairments used in different definitions. In our PCDS, we used mild cognitive impairments, MCI, by testing multiple neuropsychiatric tests. So this is our definition of PCDS. The first part, mobility impairment, MI, which includes weakness and slowness, based on the Asia Working Group for Sarcopenia, AWGS 2019 consensus. And the weakness we used hand grip strength, and the slowness we used 6 meters walking speed. In our previous study, we found that weakness and slowness had poor cognitive performance and higher mortality risk compared to non-mobility components of frailty. And the MI is an important predictor factors of the cognitive frailty. People who had MI was a good predictor of lower survival rate and poor overall health outcome. So in our definitions, we use weakness and slowness for the mobility impairment, MI. And in the cognitive impairments, we used the following 5 different neuropsychiatric tests. The first is we used the Chinese version verbal learning test CVVLT for verbal memory, and the second we used the Boston naming test BNT and the categorical verbal fluency test VFT for language, and the third we used Tyler compressed figure test for visual spatial function, and the last we used clock drawing test CDT for executive functions. And we set a cutoff line below 1.5 standard deviations with age and education match norms. And any one of these neuropsychiatric tests decline meets the cognitive impairments. And to understanding the transition of PCDS, a cohort longitudinal aging studies in Yilan, which called ELAS, was performed. We enrolled 542 subjects in second wave, which including 11 subjects become dementia in the follow-up visit. And we exclude who inability to communicate or who unable to complete the test due to poor functioning status. And we also exclude participants whose life expectancy is smaller than 6 months due to major illness. And we divided to 4 groups based on our definition of mobility impairment, cognitive impairment, and PCDS. So this is the baseline characteristics of our study. The follow-up duration is 2.5 years, and the mean age is 64.38, and the health is a man. And we also can see here the PCDS group is elderly and had lower education years than ours groups. And the second part, the physical functional assessments, we can see the PCDS and MI group had lower walking speed and grip strength. And in cognitive part, CI and PCDS showed lower verbal memory, language, visual spatial, executive functions, and a lower MMSE score. Interestingly, the cognitive function in PCDS is more severe than CI group alone. This indicated that combine of MI and CI is more severe than CI alone. And we also recorded a perpendicular skeletal muscle index, which represented the amount of the muscle. This muscle index is significantly low in PCDS and in MI group at baseline. And we also record the medical history of hypertension, diabetes, hyperlipidemia, and cardiovascular disease, which is non-significant. And this is the distribution and the transition at the first and the follow-up visit. At first, we are interested about the incidence of dementia. There are 4% PCDS became dementia, which is similar to the CI group and higher than the robust group. But interestingly, the MI group is 0% became dementia. And then the 4 group distribution at the baseline are 38, 14, 30, and 18%. After 2.5 years, the distribution changes to 20, 29, 16, and 33%, and 2% became dementia. And the lower figures showed the more details inside. The y-axis is the 4 group at the baseline, and the bar represented the second wave distribution of different groups, including robust, MI, CI, PCDS, and dementia. The CI had higher rate of transforming to PCDS than MI, which means the 26% more than the 6%. And we show that 17% robust and 29% MI and 37% CI progressed to PCDS. And the reverse rate from PCDS to robust was only 4%. Furthermore, we want to investigate factors that influence the categorical transition of 4 different groups. First, we did ANOVA and the chi-square test for each 4 baseline group transitions. For example, in the baseline MI group, the MI participant became 4 different groups after the follow-up. And we found educations, CVVLT, VFT, and CFT are significant factors between groups. And then we did multivariated binomial logistic regression models. We corrected age, sex, education, and MMSE in level 1, and we put the factors which showed significant before into the second level. And we want to find the factors affecting PCDS transition and reversion. So this is our result. The table shows the detail of the odds ratio and the p-values here. We demonstrated that in green, the robust group, who had lower CVVLT and verbal fluency test scores were more likely to progress to PCDS. And in yellow MI group, who had lower appendicular skeletal muscle ASM and elder age are easily to progress to PCDS. And in CI orange part, who had diabetes are prone to progress to PCDS. In the red PCDS group, who had a young age are easily to become MI, and who had higher CVVLT score are easily to become CI. And the most important, the PCDS with stronger high hand grip strength were more likely to revert to robust. It is interestingly that the ASM at baseline is a risk factor for becoming PCDS, and in addition, baseline higher hand grip strengths are protective factors for PCDS. The muscle function seems to play important roles. So this is our brief summary. The factors progression to PCDS include worse memory, language functions, elder age, low muscle mass, and diabetes. And the factors of reversions include higher hand grip strength, young age, and better memory functions. So the skeletal muscle mass and function are crucial factors associated with PCDS. And several studies showed that the communication between muscle and neurons. The figures showed in our previous animal model the exosomal microRNA, which is secreted from the atrophic skeletal muscle and through blood vessel to the brain, which downregulate of the neuron reactive genes and inhibit of neuron differentiations. And our study also showed irisin, myostatin, and fluostatin are also involved in muscle and neuron crosstalk. So we point out the reversibility of PCDS and indicated that controlling this risk factor of PCDS may be preventive for the disability. And this is our previous MRI studies of our PCDS. In the structural analysis, we showed that reduced gray matter in the hippocampus, middle frontal gyrus, cerebellum, and other cerebral regions. And in our diffusion tensor imaging studies, we showed that the cerebral limbic neural circuits involved and disrupted in our PCDS group. This pattern is very different to the Alzheimer's dementia related brain atrophy, which we always see in the hippocampus and the temporal lobe. So we assumed that the pathophysiology of PCDS may be different to the dementia. So our hypothesis is muscle-brain axis may be involved in PCDS. First, the muscle aging presented as a physical frailty, and our clinical data, skeletal muscle mass function, are also echoes to the muscle and the physical frailty. And through the muscle neuron communications, it can affect the brain structure and functions. So our result echoes to the muscle-brain crosstalk, and the PCDS may be the clinical consequences of the muscle atrophy, induced neuron loss in the brain, which cause cognitive impairment or dementia in our community. So we propose a novel insight of a potential muscle-brain axis involvement in the pathophysiology of our PCDS. So thanks for your attention, and thanks for our wonderful team.